We will now transition to someone else who also has been working on Kleefstra syndrome for some years now, but that from iDefine's perspective, Eric Sheff and I just recently connected with and are also very excited about the work he's doing. And so Dr. Carl Ernst is going to be joining us next on a discussion with the topic of modeling EHMT1 deficiency in human brain cells to identify new pathways for potential treatments. So Dr. Ernst, good to see you again. Take it away.
Hi, everybody. Thank you. It's nice to be here. Can you see my screen?
We can.
Okay, let me start my. So I guess first, just I would have seen or spoken to some of you families out there in Boston in 2016, and just that was a really important meeting for me. It was very nice to meet you. I'm sorry we can't be in person now. It was really nice to meet your kids then and connect with the other researchers. So thanks, Jeff, for getting this going. So I want to share, we've been working on this for about 5 years or so now and using, cells from the 9Q community. So thank you. We cannot do this without you. You're looking at a picture here of, brain cells we made from P, active cells. You can see them firing in addition. Just gives you a sense of the kind of things we can do with these cells. All right, so this is going to be a bit, I kind of want to get to my point, so I will sort of throw it at you. I will keep it as little technical as possible, but I have to go there a bit because I think we have some pretty interesting discoveries that I want to share with you. So just first off the bat, this is our population group. We have 2 patients, one of which is a full gene deletion. That's the one on the left here. And then on the right, we have a missense mutation. And this one was actually in Dr. Kleefstra's presentation. I saw it. It's in the ankyrin repeat domain. I'm assuming it's the same subject. And each one of them is matched to their sibling. And we do that just to make sure we can really reduce the variation in our experiments. So our basic approach is to make, just like you heard from Neil, and I will not repeat all that work, but we have a similar design. We make stem cells from blood, skin, or pee. We turn those into stem cells. That's sort of what you're seeing here. And then we can turn the stem cells into a progenitor-like stage. And then these progenitor cells can be turned into neurons, just as you saw from the Netherlands group. And you can do different things with these cell types. So I was very interested in, Jeff has really hinted at this today. I'm very interested in the absolute first direct effects in EHMT1 to see if we can mine down to the root cause of the disease. At some level, everything else we see is going to be a secondary, tertiary, or an indirect effect of the initial problem. So it's not to say it's not related, but it may not be the core root. So this has obsessed me for the past few years. And we did a lot of work in stem cells themselves and the neural progenitor phase, and then the neurons as well. I'm not going to share all that data with you, but we did the obvious experiments of looking at, histone modifications and, gene expression patterns. So what I want to share with you is the robust, I think, discovery we made from one of these we basically did, throw the kitchen sink at the problem, see what comes up, look for the most interesting, potentially important finding, and chase it hard. So we are very committed to trying to move this forward and get at the molecular level. So I'm going to jump right into our finding. I'll try and give a little bit of background on it, but by and large, I just want to share it with you. And you'll see, I'm really happy that you, Dr. Yamato, was before us. I wasn't aware of your paper until our results came out, but I have a model that applies very nicely to what you just presented. And I really want to stress here, this work is completely independent from the Japanese group. I was not even aware of it, but hopefully you'll see just how complementary what I'm about to show is. So our large-scale RNA sequencing of those cases and controls I showed you in the neural progenitor cells gave us a very, very robust signal. I will give some detail on this, but I just want to say that we do a lot of this across a lot of different diseases, same kind of thing you heard from the Netherlands group, where we have a lot of disorders, including Kabuki syndrome as well, and different ones in the neurodevelopmental space. So we look at a lot of this, we do a lot of genomics, and the signal we saw here was quite remarkable for this particular pathway. And this is the NFKB pathway. I will give you some information on that, but it was really, really robust. It was something we could poke around on, try and prove false, and it just kept coming up again and again and again. And I'll point out that this gene right here, this is the same gene that Dr. Yamada was just showing IL-1B from her, independent mouse studies. Okay, so what we saw is a large increase in the NF-kappa B signaling. So our immediate thinking was, is EHMT1 functioning in the cell to act as a blockade under normal standard conditions to keep this NF-kappa B signaling molecule in check? And I'm going to give you a little, I'll give you a little background on that. So just to show you just how robust these data are, here's across the patients. So here's the matched brothers from our group 1. And here's our missense patient from group 2. This is 2 females. And you can just get a sense. These are just target genes of the NF-kappa-B pathway. And this gives you an idea of the variation we're seeing. And it's remarkably tight. These are 2 different families. These are 2 different sexes. And yet you keep seeing this robust effect. Here is Dr. Yama's gene she was mentioning in the last part of her presentation. These are not expressed in NPCs, just to tell you, they're expressed in microglia, and that's what you're picking up here in the control. And yet in this condition, it is quite a strong effect. Something that is supposed to be off is being turned on. So we're really getting a sense that EHMT1 might be functioning to keep this thing in check. And again, this NF-kappa-B pathway turns on 400 to 500 different genes, which, by the way, were first discovered in the immune system. This is in NPCs, and when we look in neurons, the effect holds. So that there's a nice replication or reproduction to this. So it's the same cells, but they're differentiated, and you can see the same pathway. There's variation, but the trend holds very consistently across this. And this is also taken straight from the RNA sequencing data. So let me just tell you what this pathway is, so you can get a sense of why I'm very interested in it. So just briefly, I appreciate people are not molecular biologists out there, but it'll give you a sense of why I'm chasing this. So the NF-kappa-B are a group of proteins, they're family of related proteins that want to go and do things to the genome. They want to turn on very specific genes in the genome, and they bind a very particular sequence of bases. They need to be kept in a prison cell, essentially. They need to be stopped from doing what they're doing. And if they're not stopped, all kinds of very bad things happen. For example, there's many diseases related to this, which have interesting phenotypic overlap, by the way, with several different features of Cleistra, including heart, osteoporosis when it's hyperactive. So the interesting part about this protein is it contains this family of proteins. It has 6 to 7 anchor repeats. Now that you might be familiar with in the EHMT1 protein also has anchor repeats. Anchor repeats are a very common repeat in proteins. 3,000 to 4,000 different proteins have them, but it's the number of repeats that's critical. So the first thing that we found was interesting was that this is a potential binding partner. So what does NF-kappa-B do? It is, again, first discovered in immune function. It's known to be important in brain and can change spine density of excitatory neurons. So just to take us back to what we were hearing about from all the different speakers, what I'm trying to propose here, of course, EHMT1 could be doing many things in the cell, but this could be a very important target of what EHMT1 is regulating. And I'm going to try and convince you today that the data are very robust and it's very, very worthwhile to look into. And just to give you a sense of what I have a friend here, what the anchor and repeats look like. Here's all these NF-kappa-B family members. This is in purple. And again, these were shown in the cartoons from the previous speakers. Here's what it looks like in EHMT1. And here's the 7 repeats, these anchor and repeats. Here's our mutation in this evolutionary conserve from our family 2. So you get a sense that, and this is a well-known protein binding effect, you get a sense that binding could occur between EHMT1 and NF-kappa-B. So of course, the first thing we asked when we saw this large signal is, well, I mean, this seems like something people have shown or would have guessed before. And lo and behold, when you actually know what you're looking for in EHMT1, there's 2 very lovely papers showing interaction of EHMT1 with the main core member of NF-kappa-B, the 2 core members, and 2 independent papers. So we already have evidence that this is happening, and this never crossed my radar before because I never knew to look into this particular area. So the first thing we did was, well, do we actually see an association between EHMT1 and one of the core family members? And we did this synthetically at first, just adding tags and doing it in a little cell line, and we can see it very clearly. And when we do it endogenously in our in the little boy from group family 1, we can pick up the association in unmodified proteins, which just basically supports what the people that I just showed you on the previous slide were showing, that there is in fact an interaction, and it's probably something that we need to be very aware of. Okay, it's one thing for me to say I found this in 2 people, even if it's matched to their brother, but it'd be nice to show this in different ways. So we spent a lot of time, making cells and engineering cells. So we knocked out EHMT1, heterozygous knockout. It ended up looking like this, an insertion of 29 base pairs, which led to a loss of function. And then we asked, well, do we see the same thing, the same effect on hyperactivity of NF-kappa B? The answer is yes. So you can see here's that same pattern that we were seeing before. We have this significant increase in just the genes we picked as targets. We also did a genetic repair from the little girl from family 2. This is the 948W mutation. And I'll just, for the scientists in the audience, I'll just highlight that when we do our repairs, we always add a silent mutation just downstream. This is in the wobble position, just to ensure that we can in fact show that it is us making this change and it's not just sequencing of wild type DNA. So this is what our traces look like. And it's probably about 80 to 90% pure, so clonal. And here's what it looks like. So same thing in the neural progenitor cells versus the neurons, we see the decrease. So now we're trying to show we can rescue the effect on NF-kappa B, the specific pathway. So we're mutating the mutant EHMT1 back to healthy EHMT1 DNA, and it's lowering the effect. We're comparing it to the mutant itself. This is the only one that does not lower, but this was also our most extreme effect size if you look back at our other data. So it may still be, it's probably still having an effect there. But in any case, you can see just across the board in neurons, we're obviously interested in the neurons because this is the postmitotic state. NPCs would be more similar to an embryonic brain. So we're seeing the effect probably even more pronounced in the neuronal state. What about pharmacological inhibition? So we want to, if I want to convince you of something, I want to show you through, your own families. I want to show you through an engineered knockout that's isogenic that I've made in my lab using just healthy control cells. And I want to try and show you that if I block it pharmacologically using a known cancer drug, which I have listed here, we can actually mimic the effect of the disease. So just taking healthy cells, we add the drug, we can actually see this increase in all of these same things. It is very, very robust effect. It's unambiguous. We've picked these markers, but you could probably pick a lot of different NF-kappa B response and you're going to see it. This highlights the fact that the histone methyltransferase is probably quite important to this. It's not just the fact that it binds, but the transfer of the methyl group obviously is playing a role because this drug just blocks that. It would not block binding. I can also show you if we look at the same drug, what is the effect on some output markers. First, let me just show you that our drug is working. When we add the drug, it can block H3K9ME2, and you can see as we increase the level of drug, we get decreased H3K9ME2 exactly as you expect. So this is just to show you we're not messing things up. And then come over here and look. Here again is Dr. Amada's protein, IL-1B, which is undetectable in NPCs. It starts to be turned on, and this is protein, and we see a gradual increase in it. Here was that big marker we also saw. It's off when you have no drug, and when you start adding drug, you can, by eye, you can pick out the increase, and this is just a control. So we think these effects are very robust. We see it in the families and the patients, we see it in the engineered knockout, and we see it in just adding a drug to a dish, which is a really nice way to do things because it's an identical cell type. You don't have to deal with all the variation of the cells. Okay, this is an important, this comes up a lot, and we were surprised by this, but I was heartened to see the paper from Hans van Bakkeveen in nucleic acids research in 2016. So I told you at the beginning, we mapped a lot of histone methylation, and this is something we do a lot, and we have a lot of expertise with doing this very carefully. We did this in H3K9ME1, which is not the one people use, although I'm not exactly sure why that is. ME1 and ME2 are both targets of EHMT1. And what we saw just at the ME1 level, so this is what EHMT1 is supposed to do to the histones to do all of its effects. We looked at all the target genes. This is the level of H3K9ME1 we're seeing. And if anything, we're seeing what Hans saw, that there's a slight increase in H3K9ME1. So this doesn't seem to be any genomic effect of EHMT1 loss. This is across the patients and the knockouts on these loci. And I can tell you just across the genome, we don't see it either. Now, H3K9ME2 is in progress. Just because obviously we need to check that, given what everyone else is saying, but we really we don't see any difference here, which suggests that the effects are not happening at the genomic level unless it's happening through H3K9ME2, which it certainly could be. I will just finish up with some final data that we have to show you where I think the mechanism's occurring. We pulled out one of these core NF-kappa-B family members called RELA. This is just proof that our IP works and the antibodies working. And very interestingly, I told you at the beginning that the NF-kappa-B family, this thing that goes and turns on all these genes, has to be kept in a molecular cage. It needs to be locked down because it always wants to be active. And the role of EHMT1 is to probably help make this prison cell around NF-kappa-B. What we found when we did a mass spec, a quantitative mass spec with both patients, this is something I rarely do. My students always talk about it, but we tried it because of the sort of nature of the experiment. We were actually able to pick up quantitatively. We have a decrease in the prison cell, if you will. The proteins that create the prison around NF-kappa-B are a lot lower in the EHMT1 mutation patients. And you can see the other proteins in this family are not affected. It just seems to be the prison cell related proteins that we pick up on the mass spec. And that's again done from an IP of a separate protein. And don't take my word for it. We can even show it just on a straight Western blot. So here's our patients again. You can see very clearly, this is the prison cell protein. It is definitely depleted in the EHMT1 mutation cases. This one is our engineered. This is patient 2, the missense, and this is patient 1, the full gene deletion. It's very consistent. And then again, just to show you the control, here's IL-1B again. Just look by eye. We're just picking up this hint of IL-1B and even CXCL5 and then the loading control. So we think there's a very, very important, robust problem with keeping NF-kappa-B repressed. And NF-kappa-B could explain a number of different symptoms that children with, or anybody has with the EHMT1 loss. So just a conclusion, we think this is a new pathway to explain some of the features of the syndrome. Of course, we're in a cell right now and I'm extrapolating, but if you go and start looking into this, you can start to see the kind of things I'm talking about. We think that the dosage of EHMT1 is critical to keep NF-kappa-B in an off state, and that's the family of proteins in human brain cells. It's contributing somehow to building this prison around this transcription factor. We think it might actually interact with several of the members that have the 6 or 7 anchor and repeat domain. We just showed it in 1, but it could be more. We know that the methyltransferase domain is important from the drug studies, but we doesn't seem to be at the H3K9 methylation 1 and still could be at ME2. So we cannot rule out genomic effects yet. And then finally, I would leave you with this, and this is what we're actively trying to work on. We're trying to secure funding now for this. That overactive NF-kappa-B is a well-known, it's a targetable gene pathway that many, many drug companies have drugs for and could be a viable therapeutic strategy in EHMT1. And with that, I will leave you, and thank you for everybody for joining, and hopefully we can meet in person in the future years. Thanks a lot.